28.
Oh, I like it. Can we always have you narrate the title of the paper? Valentina, can you also share the link, please?
Also the link.
Also, yeah, no, I'm demanding this morning.
Yeah.
Oh my God. Yeah, of course.
It's just easier to share
It's
that way.
right here. Well, I shared the...
You shared the paper, but then I have to type it. If you don't
Sorry.
have it, I just grab it.
No, I have it. I have it. Now I lost Zoom though.
Romance.
Oh,
It's fine. After the last CNS talk, we can survive anything. You can lose Zoom, you can lose your presentation. We can make it work.
oh well, I'll share the link after. Now you have the PDF. You can rely on that.
Great.
Right?
Okay. Deal. Just gonna
Thank you. No, I found it. I found Zoom again. Yay. Voila.
I see Christmas jumpers. Thank you.
Oh, yay.
100 points for effort. He's not listening, I think. Here's the link for everyone over on YouTube. Right. 2 minutes delay. Let's get started. So welcome to the last Neuroccino of 2022. Today we have an exciting paper entitled "The Neurons That Restore Walking After Paralysis," and Valentina is presenting it to us. We will be back on the 9th of January next year, and it will be the same rules. Anyone who reads some exciting piece of science, papers, books, book chapters, is welcome to join us and present the paper. Now, without further ado, I can see Valentina ready to go. Take it away.
Thank you. And I just read on the chat that this is a paper from the IMN. So, but no conflict of interest, I swear. I didn't know before reading the paper. And of course, from the title, you can see why I got excited about this study. Some degrees of plasticity, so restoring walking abilities after a spinal cord injury has been documented since like the Roman times, but only with this new paper we actually individuated, so the authors individuated which neurons are in charge of this form of restoring the ability of walking. So the paper starts saying that they observed in 9 out of 9 patients the restoration of ability of walking after spinal cord injury. This restoration has been allowed by this type of device called EES. Do you see my cursor? Okay, this type of device that implants electrodes on the spinal cord, epidural part of the spinal cord of these patients, and send inputs to spinal cord neurons, and these allow the restoration of walking also after a rehabilitation program. So the implementation of this device on the spinal cord and then after like 4 weeks, if I'm not mistaken, rehabilitation program with this device that sustains the body of the patients while they perform some walking activities or activities with involving the limbs or the legs. They are actually able to recover from the spinal injury. And the patients they selected have different grades of injury, meaning that some of them have a complete differentiation of the signal, and they cannot send back sensory motor inputs to the cortex, while some other still have some degree of spare sensory motor signal reaching the cortex. And this is basically a depiction of a representation of a patient with these implants on, and that is able to walk after a spinal cord injury. In this panel here, we can see, and this is I actually find very interesting because I didn't know about this mechanism. So this is a PET scan of the spinal cord of these patients, one of these patients. And here we have the PET scan of the spinal cord before the stimulation and the rehabilitation program. And you can see this type of activation. While here we have the PET scan of the patient after the stimulation and rehabilitation program, and you can see the activation is actually lowered. And I found that this is very interesting, and we will find later on what this mechanism is. And here you have the comparison between before the stimulation and after stimulation in both metabolic activity, and statistically represented the differences in the metabolic activity, i.e. the PET scan here, and also the motor scores and a specific task, a motor task. And again, you can see after the rehabilitation, their working abilities actually improve. So the author of the study wanted to investigate which neurons are in charge of this plasticity, of this enhancing of the motor ability after spinal cord injury. And what better animal sample than the rats? So they reproduce a spinal cord injury in the rats at the level of T8. And then they implanted the simulation device, EES, also in the rats. And you here the picture shows off and on stimulation, how the rats changes in its behavior, meaning that off a stimulation, the rats is not able to move, and on stimulation, the rats restore some ability, working abilities. And they, so in these graphs here, they show the number of cell detector lower. It's just a depiction of the neurons detected in the spinal cord before and after the rehabilitation. The authors wanted to explore the neurons that are responsible of this changing, and before doing that, they created an atlas of the neurons of the spinal cord of the rats. They created this atlas by exploiting some sequences techniques of RNA sequencing of the neurons of the spinal cord. And they identified more than 20,000 neurons. And to simplify the characterization of these neurons, they performed the clusterization and saw that this neuron clusterized in 36 clusters. So these are the 36 clusters. And then they used UMAP, so a projection technique, uniform manifold approximation and projection technique, to depict, display how these clusters of neuron organized according to each other, basically in a space. It's not written if it's 2D or 3D space, but I guess it's 2D space by this image. And so this is the main representation of how the neuron organized. And they actually saw a pattern of organization of the neurons that go from the ventral to dorsal organization. And then they were able to identify, so to locate within the spinal cord where these cluster of neurons are actually set by different methodology that I don't know, I didn't understand. So I'm just going to mention it to you and showing this beautiful picture of how the neuron organize themselves onto the spinal cord. So what I did not tell you is the rats. So since, well, in animal condition, it's easier to manipulate the various conditions. So the rats went throughout 8 different conditions. So a group of rats had only the spinal cord injury. Another group of rats had the spinal cord injury and went through the rehabilitation and stimulation. And then another group of rats went throughout spinal cord injury, stimulation, and a walking behavioral paradigm just before they were terminated. And another one, the spinal cord injury, rehabilitation, and stimulation of the cortex, the motor cortex. And some other groups went throughout the combination of these different paradigms, these different conditions. Why I'm telling you this? Because then the authors wanted to see which of these neurons that have been beautifully identified were actually manipulated, so they were sensitive to perturbation induced by these conditions. To do so, they used a machine learning algorithm called Augur. I hope the pronunciation is correct. That was able to identify which of these neurons, cluster of neurons, that are whose tree is depicted right here. Is actually perturbated by the conditions, and in these from 1 to 6 are actually the contrast between the conditions. And they saw that these 2 groups of neurons, that from now on I will refer to as SC neurons, because they have so this name here, which is very complicated. And so these type of neurons were actually perturbated by throughout all the condition. And this has been confirmed by the area under the curve, referring to the comparisons between the conditions here. And on the left, they identified on the UMAP where this type of neuron cluster, and on the right, we have a depiction of how where this type of neurons cluster are located onto the spinal cord of the rats, and to reproject this type of neurons, the identification of these type of neurons onto the actual space of the spinal cord. They used a method called Magellan. So here they went through more in detail describing these type of neurons. Don't worry, I know this is a lot. It's a dense figure, but I'm trying to be brief before Steph starts pumping the sign. So these neurons have afferent connections to large diameter afferents and reticulospinal projections, and efferent connections to glutamatergic, GABAergic, and motor neurons. And basically here, just they validate this the model that they explored both in a statistical methodological way and also more biologically. But what's interesting here is that the connection, so from the large diameter afferents and the reticulospinal projections are actually modulated by the stimulation. So as we can see here, before and after stimulation, the connection increase in synapses, neurons, and also in the intensity. To conclude, and so to prove that they actually found the neurons that are responsible for walking recovery. The authors manipulate the activations of these type of neurons, both optogenetically and chemogenetically, meaning that the deactivation of these type of neurons, while the rats have been stimulated, actually modulates the recovery in walking. So you see, before they are able to walk with the stimulation, then the neurons have been silenced and they cannot walk anymore, and they recover again when the silencing has been stopped. And we see a similar pattern with the chemogenetic silencing. And to further add a control of this finding, they perform a lateral hemisection of the spinal cord that usually is followed by natural recovery of walking, but with the silencing of this type of neurons, the rats are not able to walk anymore. Do you remember the PET scan that I showed you at the beginning? Well, the spinal cord injury actually lead to aberrant activation within the spinal cord, and this type of neuron lead to inactivation of these aberrant signaling after spinal cord injury. This is why the PET scan show high activation before the stimulation and low activation after the stimulation. So I got very excited by this paper because it has so many potential outcome on the recovery after spinal cord injury in humans. I don't know much about the translation between studies from rats to humans, so I don't know how far we are to implement this in humans. And also another comment that I have is that all the paper is focused on these single neurons, like they found the neurons that restore walking. But I would like to see like further studies focusing also on the whole system, so including the connections that are responsible for the restoration in walking after spinal cord injury. Thank you for your attention, and I'm happy to discuss these points or other points that comes to your mind.
Lovely. Thank you, Valentina. I do apologize for shocking you last time for putting the timer on.
No, but you're right. I mean, we have a certain time that we have to stick to it.
I've just been signed out of my Zoom account. Well, that's interesting. Yeah, still here. Okay. Yeah, I just want to make sure that we have enough time to discuss the exciting science, and I can see hands up already. So I'm just going to hand over to Victor.
Hi, very interesting. Sorry, very interesting study. It's quite exciting to see this kind of thing, which has potential to be to revolutionize like the life of some patients and some treatments, or even just our understanding of this kind of thing. Even if it's not yet on human, you would expect that it's really the end goal here. It's really to have this translation to human. I was wondering, because if I understood correctly, these neurons that help recovering after an injury are not the neurons that were used before the injury to conduct the the information from the brain to the muscles. So do we know what they were used for before, or were just hanging around? And do you have information about that?
No, I don't have information on that. They manipulated this type of neurons in healthy rats. So to see whether ablating, silencing this type of neurons will lead to the interruption of the ability of walking in healthy rats, and it's actually not. So I don't know what they need for.
All right.
But yeah, maybe they say it somewhere in the extended data or the methods.
Okay, too bad, no problem.
But it's a good point.
Thank you.
Michelle has a question for you as well.
I think it's crazy how the plasticity in the brain is using these neurons and rats can walk again and people can walk again. And so if you project yourself in the future, and let's imagine we were doing the same thing for association pathways in the brain, and being able to rehabilitate connections that have been severed through this method of stimulation, do you think the personality and who the person is will take over, and or do you think people will be different because you change the way things are connected?
Well, again, as I was saying, here they discuss a specific type of neuron, and I don't think that personality arises from a specific type of neurons. But again, if we look at the connections that are involved in this change, I'm not saying I would expect the whole personality to change, but still it will lead to behavioral differences that in turn will lead to strategy to cope with these behavioral differences. In a higher system, like changing a gear, like a small gear, will lead to a cascade of events that eventually will lead to some changes in the whole picture. But again, as a strategy, as a cope mechanism.
Okay, so so it leads to the ethical questions. So are you able to repair the brain at any price, even though if the way you repair the brain change a person? And then it's not the same person. So the person that was there before is actually really dead.
Okay.
I don't know. I don't have the answer.
No,
I'm just curious.
I know. I'm thinking, but it's an effort of really projecting in a very distant future where you can tune all of these every neuron existing in the brain. Then I don't know. You might be right. I don't know. Extreme tuning might cause a sort of eugenetics, but after the human being born.
But then wouldn't you say that the child that you were is dead now because you're so different than him or her?
Yeah, we're talking about a completely different person, tuning all the neurons, all the.
Yeah, but you can imagine that we are actually doing that in everyday life. We are tuning all our neurons naturally through experience, memories, memory loss. That is really natural. And so the people we were 10 years ago is not the people we are right now. So is the people 10 years ago dead, or are we just It's an interesting question, but
it's interesting also relating to what you said before, because here we're using a type of neurons that are not, that don't seem not, at least as I read the paper, seem not to play a role in the normal behavior, but they are activated, so they play a role when the behavior is aberrant. So when there is a shifting in the normal behavior. So it might be that there are some other type of neurons, and it might be a coping mechanism, meaning that they are programmed to allow us to be ourselves, like also in aberrant situations. In that case, I would say it would be ethic. So to keep us in a normal, or as we are programmed, way of functioning. I don't know, but it's we are going in a very sloppy.
We're already making very big changes to some patients' brains. So I'm pretty sure that some people thought of the question already. I mean, when you're cutting a whole white matter tract, or I mean, when you're resecting a tumor, you can't only remove the tumor, so you're removing actual healthy tissue, and also with epileptic patients, if they don't respond to any treatment, the last resort is cutting something in the brain. So I'm pretty sure some people thought about that. There must be some publications.
Well,
hopefully, otherwise it's bad.
There's an entire field called neuroethics.
Yeah.
That deals with it, and then obviously precision medicine also goes in that direction. Sorry. We have glass boxes, as you can see, so it's quite distracting. But it's an interesting discussion. One point I wanted to make about what Victor said is that you're not the same person that you used to be 10 years ago. I would agree with that, but the change is slower than altering your brain through some kind of treatment or therapy. And you can direct it yourself in a way, right? You can seek out specific experiences or avoid specific things that might have an impact on you. Not all of them, obviously. But it's a slower process, and you're more in the driving seat, I would argue, than coming in for brain-changing surgery. Yeah.
And also there is not a gold standard in brain surgery. It's like on site. The surgeon see as far as it can go while removing the tumor. So I guess each surgeon has its own approach, and the health of the patient is the first goal. But in between making huge damages and removing the tumor, like slightly removing the tumor, there is a gray area that I don't think there is a gold standard for that. So if we cannot regulate this, just imagine the rehabilitation of patients that underwent a trauma.
You could take it even further. So the ethics within the medical sphere, so to say, are always guided, as you mentioned, by the benefit for the patient, right? But if you then come up with therapies that are brain altering to improve your motricity or your cognitive functions, whatever you want to throw into the mix, and then this becomes a service to healthy populations, that's a whole different ethical consideration now.
No, yeah, that's right.
Reminds me a little bit of the myth of Theseus and his ship, where Theseus went to have adventures, if we can say that, with his ship, and slowly, little by little, the ship got damaged, then repaired. And when he came back at its home decades later, there was not a single splinter on the ship that was original. It was a whole new ship in mostly the same form, but was it still the same ship that left, or was it a different ship? And if you do, it's a bit like this. And I think if you do repair brain surgery and you change a whole big part of the ship, is it still different from, is it much different from, well, it is, but conceptually, is it much different from a slow change over time? That's a difficult question, I think.
Well, that example you can take even further, because if you assume that the ship got repaired, but then the individual pieces of wood get just let go into the water, and then someone picks up the individual pieces and rebuilds the ship.
Yeah, I know.
It's a nice thought experiment. And you're not the only one who thought about it.
I know.
See in the chat.
So Pascal, I think, mentioned that. So there was a paper in Nature like 20, 30 years ago. I was not reading scientific papers at that time. So I'm not that old. That like claimed that a bird that was like extracted and put like with a different population of bird, because all these cells are like replaced over time. I think in a year or 2 years, it's an entire new bird because all the cells have been replaced. This bird is completely acclimated with a new population of bird because it's an entirely new bird. And Pascal Reichenbach went to present and criticize this paper in this big meeting and said, I've been in the United States. He's like an East European researcher with a very thick accent. He said, I've been in the States for more than 20 years. And if that story was true, I wouldn't have that accent. So somehow you can replace your cells, but still keep the entire essence of who you are, despite being completely different. That was his idea. Damn, I killed the buzz. I'm sorry.
No, it's fine.
I think you got people thinking here. But we're also out of time. So as much as I hate to interrupt this discussion, it's time for us to let go, say goodbye. Thanks for joining us for the last Neuroccino of 2022. And we'll be back on the 9th of January, I think, but do check online. We'll announce it. Bye-bye, Mish. Bye, everyone. Have happy holidays. See you next year.
Bye.
Bye.